This class we will discuss about the hematopoiesis. So what is the mean of it? So hematopoiesis means the formation of the blood cells. So in blood we have already discussed the types of cells are there, like RBC, WBC, like white blood cells, platelets along with the NK cells, T cells, B cells are there. So how these kind of cells are produced, formed in that blood, so that we will discuss in that entire chapter. So from starting with the first, how it will be coming. So for we all know, we in that zygote condition. Okay, let's going from that part. This is a single cell we called the zygote. It's a 2n. When it is divided, this is the stem cell. So zygote is a stem cell. It's called the totipotent stem cell. So in division, it will divide into that will be the equal division. It means cell do not lose their potency. So it's a totipotent, written down, totipotent. That means it can generate the complete body, that is called the totipotent. So these are the totipotent stem cells. So first, the zygote to the equal division is there. That means cell is divided into two, and in stage to 2 cell to the 4 cell, 4 cell to the 8 cell, and after 8 cell, morula stage is there and blastula stage is there. So now we are coming to the blastula stage, whether one ball like structure will come, and the upper layer is known as the trophoblast. This is called the trophoblast. These are the trophoblast. These cells. So inside of it, few inner cell mass is there. This is the inner cell mass. So these are the trophoblast, zygote to inner cell mass in blastula stage. So we called it ICM, inner cell mass. This in that condition, it turns to the totipotent to the pluripotent. So in zygote stage to that blastula stage, it turns to the pluripotent. Pluripotent, it means it does not have the entire formation. The inner cell mass don't have that complete body formation, because trophoblast is get separated or differentiated from the totipotent. So now we got the pluripotent cell. So our target is the pluripotent cell. So now I am going to the next slide. Yes. So written down in yellow, we have the pluripotent IC inner cell mass is there. So this inner cell mass is now controlled by the 2-3 genes like RANX1 or AML1. This kind of regulatory genes that changes the expression. So now the pluripotent cell is get changed into two. It means it's differentiated into two. One cell is the HSC, hematopoietic stem cell, and another will be the mesenchymal stem cell. So one will be the HSC, and by the control over the RANX1 gene and AML1 gene, and it gets changed to the HSC. Another one will be the MSC or MSC, that is the mesenchymal cell. These HSC get changed to the, in brief I can just write down it. No, I will just make it clear. This HSC, like two things happen. One is the self-renewal, because it's a pluripotent stem cell, so it needs the self-renewal. First thing, it needs the self-renewal. For self-renewal, if needed the self-renewal, one major gene is responsible, that is the BEAM, that is the BEAM1 gene. BEAM1 gene is responsible for the self-renewal of the hematopoietic stem cell. So it's the same cell. If I take yellow, it is a same cell, self-renewal. It's like one part of the renewal, it's completely depend on the BEAM1 gene. Now another portion is controlled by two genes. One is controlled by the IKROS transcription factor. IKROS transcription factor that mainly responsible for the lymphoid tissue. So one differentiation changes the lymphoid. Another will be the myeloid. For myeloid, the major gene is the GATA, GATA1, and it changes to the myeloid. One will be the lymphoid differentiation, another will be the myeloid differentiation. So in brief, now the transcription factor. First, the transcription factor I have discussed, the RANX1 and the AML1, and that involve the pluripotent inner cell mass to the hematopoietic stem cell and mesenchymal stem cell that can differentiate. And after the hematopoietic stem cell to the lymphoid stem cell and myeloid stem cell, that involve the 3 or 4 lot of transcription factors are there. I have just discussed the major one of the transcription factor. The first, the BEAM1, the major transcription factor that involved for the self renewal of the hematopoietic stem cell, and another will be the lymphoid stem cell that controlled by the IKROS lymphoid stem cell, and if GATA1 is responsible for the myeloid stem cell. Because we are studying the hematopoiesis, it means all the blood cells get formed. So in that picture you can show the myeloid progenitor stem cell and lymphoid progenitor stem cell. From the myeloid progenitor stem cell, the erythrocytes, platelets, eosinophil and the neutrophils are there, and from the lymphoid progenitor stem cell it would be T cell and B cell and the NK cells. So So first the clear differentiation should be there. From that differentiation we get the GATA and myeloid. So GATA and Icaros is the transcription factor, and the myeloid and the lymphoid. Now I go for the lymphoid part. So further differentiation, uh, give that one will be the dendritic cell, another will be B cell, other one will be T cell and NK cell. For myeloid one, let's take the red color to be prominent. Yes, for myeloid cell there are different kinds of another transcription factor. Just we remind it very minutely. One will be the GCSF and the MCSF. For GCSF it will be the myeloblast, because it gives the granulocytes. So it's you can write down myelocyte on photo will be mine there. Okay. For myelocyte it can differentiate it into the, if we write them down it will be better. Okay, next page. Okay, now let's see. Granulocyte. So from granulocyte we know what are the key components. One will be the neutrophil, one will be the basophil, and another will be the eosinophil. So neutrophil, basophil, eosinophil, that is the granulocyte, and for GCSF that